In the previous video, we covered some of the major organelles of the eukaryotic cell. This is from Study Guide Worksheet number 2, and this is kind of the last piece of Unit 2 over the eukaryotic cell. And so in that earlier video, we looked at the nucleus and the nucleolus and the rough and smooth endoplasmic reticulum, and we looked at the ribosomes. And so that's the end of video part 1. We kind of finished up here with the ribosomes. We're going to jump in and get the second half of this study guide worksheet number 2 and finish up unit 2. So flip this. And at the top of the page, this should start with the Golgi apparatus. And so we will basically draw in the structures that we had previously. And we'll start with a plasma membrane, and then we'll draw in the nucleus. Some of these structures that we labeled earlier, I'm not going to go back just for the sake of time and relabel those. You should have those labeled on your earlier graphics. We know that we have the two of the endoplasmic reticulums, the rough ER and the smooth ER, and the difference being the presence of the ribosomes on the surface of the rough ER. So we can go ahead and draw those in. And we also had some ribosomes out here, these free-floating ribosomes. The Golgi apparatus is named for Antoine Golgi, and based on the name you might guess his country of origin is Italy. And the Golgi apparatus basically is this kind of series of membranous tubules. And we've talked kind of briefly earlier about all of these organelles kind of performing specific functions for the cell. And there's been that comparison made, or that analogy made, of the cell being like a factory. If the cell was a factory, or if you had like different roles assigned to each of these organelles, the Golgi apparatus to me is like the UPS or the FedEx of the cell. Essentially, it receives, modifies, stores, and packages proteins for export out of the cell. And so here is, we'll go ahead and label this just because we haven't seen this one yet. 2 P's in this one, a bunch of A's, and just 1 R. So that's the Golgi apparatus. I don't know, we'll slide over here and get the function for this real quickly. Basically, it receives proteins that are coming from the ribosomes on the surface of the rough endoplasmic reticulum. It may modify those proteins, so it might change the shape or the structure, the conformation. It might add a sugar to it, but then it will temporarily store those proteins, eventually package them for export out of the cell. So again, to me, it's FedEx, UPS. It's kind of the shipping center for the cell. It has a receiving side and kind of a transport side, but the function of the Golgi to receive proteins from the rough endoplasmic reticulum, modifies, stores, packages these proteins for export out of the cell. That brings us to this whole series of basically storage organelles: vesicles, lysosomes, vacuoles, and plastids. And the general shape or structure for these storage organelles, you can't really tell what the organelle is unless you know what's inside of these storage organelles. But these are sac-like storage organelles. with a single membrane. That W with a slash means width. So it's hard to know which of these storage organelles you're dealing with unless you know what the substance is inside of the organelle. Now this single membrane, that's true of all of the storage organelles, but there is an exception. And that exception are the plastids. And when you hear plastids, we'll talk about these here in just a minute, but the plastids, especially the chloroplasts, they actually have a double membrane. If you think back to the origins of plastids is, explained through the endosymbiotic theory, then, that kind of gives you basically a point of reference for why these particular storage organelles may have an additional membrane associated with them. The specific function for vesicles, they are the storage organelle for proteins for the cell. So they store proteins until they're exported out of the cell. They are derived from the Golgi apparatus. So the vesicle is the storage organelle for proteins for the cell. Down here, we're going to take a look at how these proteins move. Basically that are made on the ribosomes on the surface of the rough ER as they're kind of moved out of the cell. We could do this through a series of drawings, but I think if we make a large enough cell here, maybe we can get it done in one graphic. And so again, back to this kind of same old drawing of a eukaryotic cell. Include all of the organelles that we've talked about up to this point. The nucleus, the nucleolus, smooth and rough endoplasmic reticulum. Make that ER rough, basically by adding the ribosomes on the surface of the rough ER. And then go ahead and draw in your Golgi apparatus. We'll probably draw this just a little bit bigger than we did before. When the proteins that are synthesized at the ribosomes on the surface of the rough ER, they are temporarily stored kind of in those membranous tubules, but eventually those proteins will be transported to the Golgi apparatus. And so this is the Golgi apparatus. As those proteins come into the Golgi apparatus, they actually move into the Golgi apparatus. And so you have this protein kind of being taken in, maybe modified, temporarily stored, but eventually it's going to be packaged. And when it's packaged, a piece or a part of basically that Golgi apparatus develops into the vesicle. And so this would be a vesicle that's containing a protein. That's packaged for export out of the cell. You remember that the major function of the plasma membrane is to control what comes into and out of the cell. So we have to be able to get this protein out of the cell without compromising the integrity basically of the membrane. And the way that happens is this vesicle will fuse with the plasma membrane, and then you have the protein kind of inside of there, and you're able to maintain the basically the integrity of the membrane, and then this piece of the membrane will break down, and then the protein will be released outside of the cell. So that's kind of how proteins move through this process of, once they are made at the ribosomes on the surface of the rough ER, transported to the Golgi apparatus, and then eventually exported out of the cell. These are some other examples of these storage organelles. And the first one that we'll talk about are lysosomes. Lysosomes are kind of like basically the cell's recycling system. And so the lysosomes contain digestive enzymes that will break down basically old or damaged organelles and recycle those molecules. And not just old organelles, but also maybe molecules that are, compromised or already in the process of being broken down. So the lysosomes are found only in animal cells. And I'm sure that there's an analogy to the lysosomes in plant cells, but lysosomes, and if you kind of reflect on how plant and animal cells make their living, animals need kind of more of an internal digestive system to break down some of those worn out organelles and molecules, and they're not, just disposed of, they're recycled and they're reused by the cell. So those are the lysosomes. Vacuoles store variable substances, and they are found in both plant and animal cells. The substances that they store might include like excess water, or nutrients, or maybe even waste before it's exported out of the cell. These, again, are found in both plant and animal cells. They can actually be a huge part of the interior of plant cells. And so in plant cells, if you see some of these diagrams of plant cells, you might see 60-70% of the interior of plant cells being occupied by those vacuoles. They play a really important role in terms of storing water and nutrients in plant cells. The plastids store pigments, and pigments are basically colored molecules. The one that we're most familiar with, obviously, is the chloroplast, but there are others. And so sometimes you'll see different colors in plants, the oranges and yellows and, reds and things. Those are types of pigment molecules that can be stored in different types of plastids. But the one we're most familiar with, obviously, is the chloroplast. And so we'll put this up here. And the chloroplast primarily store chlorophyll A and chlorophyll B, and we know that those are involved directly in photosynthesis and generating energy source for the plant. The vesicles, which of these organelles would generally be found in plant cells? Everything except lysosomes. So you would have vesicles, vacuoles, and plastids. And then the ones that would be found in animal cells, it's basically everything except plastids. So you have vesicles, vacuoles, and lysosomes. We get a quick look maybe at the whole page here and give you just a second in case you want to go back and pick up on anything. So that's the Golgi Apparatus and the storage organelles. We're getting close. We just have one organelle left. Well, I guess one major organelle and then a couple of organelles associated with movement or motility. So on the next page, should start with mitochondria. And remember, the mitochondria is the energy organelle. I'm not going to draw up the whole cell here. We'll just kind of cut to the chase and draw up a mitochondria. A mitochondria is kind of this oblong shape with the outer membrane. And the mitochondria has, it's a double membrane. And so this is, I'm going to call it OM. That's the outer membrane. And then on the inside, you have an inner membrane that's folded back and forth on itself. And that folding of that inner membrane really increases the surface area and allows kind of a larger area to be available for these chemical reactions that are being performed by the mitochondria. That inner membrane is called the cristae. And this is where the chemical reactions are occurring that support cellular respiration. This area or this space inside of the cristae is called the matrix. And the matrix basically is used for storage for the molecules that will be used in this chemical reaction of cellular respiration. So the mitochondria, basically, what its function is it breaks down glucose in the presence of oxygen and generates ATP. So the function of the mitochondria is it breaks down glucose. Remember that 6-carbon sugar in the presence of oxygen. Remember that oxygen kind of exerts a strong pull on those electrons in the glucose molecule. And as those electrons are being shuffled from glucose to oxygen, the energy that's being released is used to generate ATP. Which we've talked a little bit about before, that ATP is the the primary, ultimate energy source for driving chemical reactions in all living organisms. So how's the number of mitochondria determined for the cell? It's really based on the energy needs of the cell. And so, for example, some of your cells don't necessarily need a tremendous amount of energy, your bone cells and once you're mature, but some cells will need a lot of energy. And so like your striated muscles or skeletal muscles, it's doing a lot of work, they will need additional mitochondria just because they have higher energy demands. So it's variable, the number of mitochondria per cell, and it's based on the energy needs of the cell. And the molecule that we've been talking about, adenosine triphosphate, it asks you to draw kind of a diagram of this. We're not going to do a real detailed diagram of this, but we start with adenosine, which is kind of the major component of the molecule. And you may have heard adenine before, and that's one of those nitrogenous bases that we find in DNA. So some of these molecules, the cell's really efficient in terms of using, a same molecule or similar molecule for different functions. So that's the adenosine part of it. Tri obviously is 3. That means that you have 3 phosphate groups attached to this. So those are the first 2 phosphate groups, and then these are just the bonds. Notice that this terminal phosphate, the 3rd phosphate, has a high energy bond associated with it. This is actually a high energy bond as well, but when the cell needs or uses energy, basically what it does is it cleaves, or it takes off that terminal phosphate. It's kind of like releasing the energy from a battery, and it will attach that terminal phosphate to another molecule, and it kind of energizes that molecule and provides it a little bit of a jump start in order to kind of induce it or get it to engage in a chemical reaction. And so that's the ATP, adenosine triphosphate. Last thing that we have basically is to draw some of these organelles that are associated with movement. And there are 2 of them, and the difference is basically numbered based on numbers and size. And so if a cell has these extensions basically of the plasma membrane, these little hair-like extensions, and there's a bunch of them, that is the cilia. And not all cells have this, but some do. So for example, like the cells that line your trachea that help to keep dust and particulate matter from entering your lungs, they will have these cilia attached to them, and they'll produce mucus and help to kind of capture that and prevent it from entering into your lungs. So cilia on some cells, and the other thing that cilia will do is, for example, in some protozoans, it will actually provide motility to the cell, some of these unicellular protozoans. The other organelle that you can see that's associated with motility or movement is the flagella. And cilia are many and small, and flagella are few and large. So you may just see one flagella associated with a cell, or you may see 1 to 2. And again, probably the best example that we're familiar with, if you look at it, is the gamete or the reproductive cell in males, which is the sperm cell. But you also see flagella again in these unicellular protozoans, things like the euglenozoans that we had discussed earlier in eukaryotic cells. 2 functions of the cilia. Basically, it moves the cell through a water-based solution. So it can move the cell around, or it can move the solution around the cell. So it can move water or a water-based solution around the cell. Some protozoans will use this as kind of like a feeding mechanism. And so like paramecium and blepharisma, they'll actually kind of generate a current or move that water-based medium around the cell and then kind of capture any particulate matter that's suspended in that solution. For the flagella, it's only got one job. It's basically drives and provides energy for movement of the cell. And so the flagella moves the cell through a water-based solution. It's kind of interesting to watch the difference in terms of how these protozoans will move, if it's a ciliate or a flagellate. The ciliates, they can kind of move basically. They have like lateral movement, and they can move up and down and side to side and pretty much in 360 degrees in any direction. So they'll kind of change directions at any time. The flagellates, they tend to kind of just plow ahead in one direction. And so at the microscopic level, you can even tell a little bit about, what type of cell you may be dealing with, or whether or not it's a ciliate or flagellate, just based on the type of movement in those cells. That finishes up really all of the major organelles. If you'll look at the last part of this study guide worksheet number 2, there's a series of multiple choice questions. I'm not going to do those for you today on this video. I'd like for you to do those on your own. And then there's actually a total, I think, of 19 of these. There's front and back. These questions won't necessarily be exactly the same questions that you will see on the Unit 2 test, but they will be similar in terms of content. So use your notes, use the information that you have from the PowerPoint presentations, the videos, the book, all of the above, and do your best to kind of answer these questions. And we will do, if you need help on these, or if you have questions related to the last part of this study guide number 2, then we can cover that through a Zoom session, or send me an email, and we can take care of it that way. So that concludes the information for Unit 2, and so pretty much wraps everything up on eukaryotic cell structures. Thanks.